The anorectic effect of neuropeptide AF on satiety-related hypothalamic nuclei by Lake Dean and Rebecca Walko, Group 170. Appetite is regulated by the interactions of a complex system and central peripheral signals that influence individual responses to ingestion. Peripheral regulation consists of satiety and adiposity signals involved with the hypothalamus and caudal brainstem, while central control involves neuropeptidergic, monoaminergic, and endocannabinoid systems. Originating from the gastrointestinal tract, the satiety signals CCK, GLP-1, and PYY can be seen at the bottom of the second diagram, reach the nucleus tractus solitarius, also known as the NTS, through the vagus nerve during ingestion. Once the satiety signals reach the NTS, they are integrated with adiposity signals by afferent fibers that project from the NTS to the arcuate nucleus. A complex network of neural circuits is thus produced and responsible for an individual's response to ingestion. Neurons in the arcuate nucleus produce peptides that are involved in both appetite stimulation and suppression. The secreted peptides NPY and AGRP stimulate appetite, while POMC and CART suppress appetite. These proteins then project into the PVN and LHA, PFA, and induce or suppress appetite. RF amides are a family of neuropeptides consisting of a common carboxyl terminal arginine and amidated phenylalanine motif. There are 5 groups of RF amide peptides, one of which is known as the NPAF group, and is comprised of NPFF and NPAF. Both are encoded by the FARP1 gene and expressed in the hypothalamus and nucleus of solitary tract. They were originally purified from bovine brain extracts. NPFF is associated with water balance, locomotion, reward, modulation of opioid pain and function, reward and food intake in rats and mice, body temperature, improvement of spatial memory, which is only in low doses. Neuropeptide AF, also known as NPAF, is an amidated octopeptide that acts through the G protein-coupled receptor NPFF1. Little is known about the RF amide, but experiments have shown that involvement in physiological functions of the CNS. An examination of the involvement with passive avoidance learning, NPAF demonstrated improvement and consolidation of passive avoidance learning through variance neurotransmitters and a reversal of B-amyloid 25 through 35 induced memory impairment. In another study, NPAF was found to induce anxiety-like behavior in mice. Many of the RF amides are associated with feeding. Specifically, NPFF has been previously found to induce anorexia in mice and chickens. Because of this association, it was proposed that NPAF may also be associated with anorexia due to its similarity with NPFF. Additionally, it was found that the highest immunoreactivity of NPAF is in the hypothalamus, which is the area associated with the regions of hunger and satiety. This only reinforces the idea behind the concept. Targeting the left lateral ventricle, 500 newborn cob chickens were injected with 5 microliters of NPAF that had been dissolved in artificial cerebrospinal fluid along with Evans blue dye to confer localization of the injection site. In the first experiment, the chickens fasted for 180 minutes and were then randomly injected with either 0, 4, 8, or 16 nanomoles of NPAF by ICV injection. The chicks were returned to their cages and food intake was monitored every 30 minutes for 180 minutes. Trunk blood was then collected and blood glucose concentration was determined. The same procedure was followed for experiment 2, with the exception of the chicks being water restricted prior to injection and being restricted from food following the injection. In experiment 3, the chicks fasted for 180 minutes and were then given 0 or 8 nanomoles NPAF randomly via ICV injection. 30 minutes following the injection, the chicks were anesthetized and decapitated and their brains were sectioned using a cryostat in order to determine the number of reactive cells for each nucleus through the light microscopy. In experiment 4, the chicks were kept in individual cages and fasted for 180 minutes. They were then randomly assigned to receive 0 or 8 nanomoles of NAPF. After injections, chicks were placed in a recording arena with food and water containers in diagonal corners and recorded from 3 different angles for 30 minutes post-injection. Their behavior was then analyzed using any Maze behavioral software. In determining the effects of NPAF on food intake, it was found that the higher the concentrations of NPAF given to the chicks, the higher the reduction of food intake. As can be seen by the first graph, the amount of food consumed by the chicks injected with 8 and 16 nanomoles of NPAF was very similar, while the amount of food consumed by the chicks that received 0 and 4 nanomoles of NPAF was also similar. As from the time of injection increase, the difference in cumulative food intake between the chicks injected with 0 and 4 nanomoles of NPAF mostly remained consistent, while the difference in cumulative food intake between chicks injected with 8 and 16 nanomoles of NPAF decreased as from the time of injection increased up until 120 minutes post-injection. when instead of leveling out, the cumulative food intake of the chicks injected with 16 nanomoles NPAF actually surpassed that of chicks injected with 8 nanomoles NPAF. In determining the effects of water intake on the chicks, it was also found that water intake was reduced with the injection of NPAF. Water intake was reduced and most significantly for chicks with 16 nanomoles of NPAF injection and was reduced more in chicks injected with 4 nanomoles NPAF than chicks injected with 8 nanomoles NPAF. In determining the effect of NPAF on blood glucose concentration, it was shown that blood glucose concentration was reduced for all doses of NPAF. In determining the effect that NPAF would have on water and food intake in chicks, it was found that water intake was reduced by the injection of NPAF. The chicks injected with 8 and 16 nanomoles of NPAF experienced reduced water intake compared to the lower doses up until 120 minutes post-injection. Though not evident from the graph, the following 120 minutes post-injection, there was a reduction in water intake for chicks with the 4 nanomoles NPAF in addition to the chicks injected with 8 and 16 moles NPAF. Another experiment was conducted, and this time observing the CFOS levels in chicks who had been given intracerebroventricular injections of NPAF. It was found that the chicks who were given the injection had significantly increased CFOS levels in the DMH, PAMC, PAPC, and the VMH. Additionally, it should be noted that there were no effects on the LH from NPAF. Experiment 4 involved observing the resulting behaviors of chicks who had been injected with NPAF and those who hadn't. It was observed that chicks in the experimental group pecked less at food and jumped less in a 30-minute window than compared to the control chicks. The chicks who had NPAF injections also spent more time sleeping and resting than the control chicks. Many other ordinary behaviors, though, were not affected by the NPAF injections, such as preening, sitting, and standing. The most dramatic behavioral difference between the two groups, however, was the significant decline in appetite and thirst in the NPAF-treated chicks. The data supported the driving idea behind the study that the appetite-related effects of RF amides may be conserved because many members of the NPFF prolactin-releasing peptide family also affect feeding. The results all pointed to the fact that by injecting neuropeptide AF into a chick, it would cause short-term anorexia. The question still remains, though, as to how the physiology behind the cause and effect relationship works. It is known, however, that the bulk of the physiology changes occur in the hypothalamus, which is thought to mediate hunger and appetite. The hypothalamic chemistry is specifically altered within the DMN, PAMC, PAPC, and the VMH. In all these areas, the study shows a significant increase in CFOS production from NPAF injections. Because CFOS facilitates the transcription of genes, it suggests that some unknown protein is being transcribed in these hypothalamic nuclei which cause the anorexic behavior. More research should be done to investigate the biochemistry behind this transcription. Additionally, the study suggests that NPAF is better at having a sustaining effect on the body's food intake mechanism. This would be useful if a weight loss drug or medicine was applied to NPAF. It would have a much longer lasting appetite regulation effect than NPFF would. The chicks also had a decrease in thirst and water intake. This behavior is different than most other neurotransmitters and peptides that also inhibit hunger. The lack of thirst could imply that the chicks were retaining more water than usual, or even that the addition of NPAF made the chicks feel ill. Further investigation into this matter should be looked at. To conclude, NPAF injections caused a reduction in whole blood glucose concentration, increased the number of CFOS immunoreactive cells in the dorsomedial, paraventricular, and ventromedial nuclei. The NPAF-treated chicks ate less, drank less, spent less time perching, and more time in deep rest, and we can also conclude that it has anorexic effects on chicks and a direct effect on the hypothalamus. This study provides an abundance of insight as to how these neuropeptides affect chicks. However, there are a few critiques. To really conclude and apply these findings to other mammals and humans, more tests need to be conducted on various animals. This is because although the two investigated RF amides cause short-term anorexic behaviors in chickens, they may not do the same with other animals. Animals do often not have the same appetite regulators, which can be attributed to evolution. Thus, tests on many species need to be conducted to say for sure how these proteins would affect other animals as well as people. There are major applications for this study. One major one is the findings that these could have in the weight loss industry. Weight loss pills and medications are always booming, and NPAF could hold high potential for becoming a new product with minimal side effects to help people lose weight and reduce caloric intake. Additionally, the study showed that chicks that were injected with NPAF lowered their blood glucose levels significantly over a sustained period of time. If testing was conclusive that this effect would take place in humans, then implications of a drug could be made using NPAF that could not only affect weight loss, but also control diabetes and blood sugar levels as well. Further studies should be conducted to discover how this peptide affects humans and other organisms, how it controls appetite and possible side effects, regulates blood glucose, and also how the neurophysiology truly works.